Good evening and welcome to the Friday night lecture. It's a real pleasure tonight to introduce our Glassman lecturer, Dr. Jeff Lichtman. Jeff is the Jeremy R. Knowles Professor of Molecular and Cellular Biology and the Santiago Ramon Cajal Professor of Arts and Sciences at Harvard University. It is particularly appropriate that Jeff holds the Cajal Professorship. In 1906, Cajal and Golgi were awarded the Nobel Prize in Physiology or Medicine for their seminal studies of the structure of the nervous system. Cajal's careful observations of neuronal structure led to foundational discoveries in neuroscience. In fact, to the very discovery of the synapse itself. Just as importantly, his gorgeous drawings have inspired generations of scientists to delve into the mysteries of the brain. And if you haven't done it yet, I encourage you to go to Boston and see those drawings for yourself. Jeff's research represents the 21st century incarnation of the Cajal tradition. 3 words can sum up his scientific approach. Seeing is believing. Jeff's fascination with visualizing the natural world began at an early age. In fact, he owned his first Leica microscope in 2nd grade. Over his 30-year career as a neuroscientist, Jeff has sought to understand how the nervous system changes as animals develop and age. To do so, he and his colleagues have developed new microscope technologies and biological tools to interrogate cellular connectivity with exquisite detail. Among his most remarkable inventions, from my perspective, is that of the rainbow mouse. This has been widely adopted not only by neuroscientists, but more broadly by physiologists to visualize the brain and other complex tissues in a kaleidoscope of colors. Most recently, Jeff has tackled the daunting challenge of mapping entire neural networks via automated electron microscopy approaches. In addition to his contributions to both technology development and scientific discovery, Jeff's career is notable because of his sustained commitment to education, particularly here at the MBL. He has served as a trustee of the Grass Foundation and co-director of the neurobiology course, which I can tell you is quite a big job. Moreover, he has taught in the summer courses at the MBL for the past 25 years, every year for 25 years. I had the pleasure of attending Jeff's 2-day lecture on principles of light microscopy, which kicked off our neurobiology imaging section this week. The clarity with which he presented quantitative aspects of optics was simply awe-inspiring, both to the faculty and to the students. As you're about to experience, he is a terrific teacher. So please join me in welcoming Jeff to the podium.
Thank you, Ellen, for that great, kind, generous introduction. I am very grateful to be here and grateful to share with you some of the work that my colleagues and I have been doing. I'm going to be talking about one particular organ system, but I think all of you don't have to be a biologist, all of you probably know that all animals, especially including us, have a number of vital organs. And these are called vital organs because you cannot live without them. Liver, lung, kidneys, the brain. And despite the fact that we all have all of these organs, I think we have a special, let's say, chauvinism towards our brains. I'll give you an example. If your doctor tells you your kidneys are shot and he's afraid you're going to need a kidney transplant to save your life, you'd probably say, okay, that's a good idea. The same even if you needed a liver transplant or a lung transplant or sometimes even a heart transplant. But if your doctor said to you, your body's holding up pretty good, but your brain, not so good. We could put someone else's brain in your body. What do you think? I think most of you would, like me, say, I don't think so. And why is it that we hold our brains more dear than these other organs is pretty obvious. It's that The brain is where we store all that information about the life we've lived. All of our experiences, all of our knowledge are built into our brains. And it is the only organ we have that is associated with our personhood. Your skin, not so much, definitely not your kidney, livers, or lung, but your brain is you. You are your brain. And this raises an important question. How does this piece of tissue in your body store all this information about the world you grew up in, all the knowledge you have? How is that done? No other organ has this kind of memory of your life. And in what form are those memories? You know, everyone here knows what the American flag looks like. That means that somewhere in your nervous system, it's wired in, it's built in some way. It's instantiated, if you will, using a fancy word, into your brain. But how and where? What is the American flag in your brain, or an image of your mother? In what form is it? Does it weigh something? What is a memory? I'm embarrassed to say that despite neurobiologists' efforts to understand brains, we still don't have a very clear idea of how information is stored. The other thing is, not only do we store this information, but we hold it very stably for a very long time. If you're lucky, you may even have memories that last 100 years. Most of us, unlikely, but a few of you probably will have memories that will be 100 years old. How can a memory be stored so stably? You know, nowadays, I think the emphasis is often on plasticity, the changing brain. But the real magic, for me at least, of memory is that you can place a memory in there, and if you don't reconsolidate it multiple times, you just have it there, you don't use it, it just sits there, and it can sit there for decades and decades. And we probably go to our graves with many of the memories that we've made never coming back into livelihood, largely because nothing triggered them. So this is a deeply interesting question, and one that I'm quite interested in. I'd like to actually emphasize by beginning by showing you some very important data that came from YouTube, where I get most of my information now, of ways in which stability of the nervous system is evident, and also the cost of that stability. The first video I'm going to show you is one about motor systems. That's not cars, but the actions of human beings, and how once we learn something, that kind of knowledge is hard to erase. So let's start with that video, and then I'll show you one on sensory systems and one on multimodal systems. I'll get to each of them. So let's start with the first one.
With a MacGyver mullet, I learned how to ride a bike when I was really young. I had learned a life skill and I was really proud of it. Everything changed though when my friend Barney called me 25 years later. Where I work, the welders are geniuses and they like to play jokes on the engineers. He had a challenge for me. He had built a special bicycle and he wanted me to try to ride it. He had only changed one thing. When you turn the handlebar to the left, the wheel goes to the right. When you turn it to the right, the wheel goes to the left. I thought this would be easy, so I hopped on the bike ready to demonstrate how quickly I could conquer this.
And here he is, ladies and gentlemen, Mr. Justin Sandlin. First attempt riding the bicycle.
All right. So the faster I go, the better.
Yeah.
I couldn't do it. You can see that I'm laughing, but I'm actually really frustrated. In this moment, I had a really deep revelation. My thinking was in a rut. This bike revealed a very deep truth to me.
So that bothered him, and so he decided to master this bicycle. And so I'm going to skip the in-between and let him tell you what happened 8 months later.
But after 8 months, this happened. One day I couldn't ride the bike, and the next day I could. It was like I could feel some kind of pathway in my brain that was now unlocked. It was really weird though. It's like there's this trail in my brain, but if I wasn't paying close enough attention to it, my brain would easily lose that neural path and jump back onto the old road it was more familiar with. Any small distractions at all, like a cell phone ringing in my pocket, would instantly throw my brain back to the old control algorithm and I would wreck. But at least I could ride it.
So it took him 8 months, and he did it, but that most of us wouldn't spend 8 months learning to ride a bicycle as an adult. he had these welders also make a small version for his son, who was 6, and it took his son a week and a half to learn. So there's a message there, which we'll come back to. I now want to show you a sensory kind of brittleness again, where you can't change the way you see the following thing, even though intellectually you know what to do. And this is a lecture, part of a lecture given by Richard Gregory, the late Richard Gregory from the University of Bristol, is a neuropsychologist, explaining, if you listen to what he says, why it is so hard for us to break out of this optical illusion. We should be hearing this.
Normal head of Charlie Chaplin, but wait, as it comes round, you'll see, or will you, that it's hollow. The back of it, coming round now, is actually a hollow mask. It appears to rotate in the opposite direction, and amazingly, the nose sticks out, although it's actually sticking in. Coming round now is the normal, correct as it were, face, and wait again as it comes round, and you'll see this extraordinary thing like Jekyll and Hyde. Both the noses stick out because it's so unlikely that a nose sticks in that a face is hollow. So you see it as convex, although it's in fact concave, as now, and then it'll become the normal face again, there. And note that as soon as the features appear in the hollow inside, it will look convex, as though it's a normal face almost, though it isn't. As soon as the features appear, there, your brain refuses to see it as hollow, simply because it is so unlikely. And this demonstrates the immense power of top-down knowledge, which can actually counter signals bottom up from the senses and force an extraordinary illusion in which the sensory information of the present is cancelled by immense knowledge derived from the past, because you've seen so many faces, all with their noses sticking out. So it's just impossible to see that as correctly hollow.
So the last illusion is more subtle, but it is spooky, like magic for me. The person who's going to be showing you this illusion is actually a magician, Mark Mitton, but this is not magic. This is your brain fooling you. He is going to be saying throughout the video, I'm going to show you one syllable. It's the syllable that black sheep make, ba, B-A, ba. That's all he's going to say. But if you watch him, what he sounds like he's saying will change. And if you doubt what you're hearing, just close your eyes, and you will hear it's just a ba. And the last part of this video, there's going to be 3 versions of Mark, and which one you look at will determine what you hear. And that means you can't trust what's coming in your ears. And by analogy, maybe you can't trust anything. So this is a very famous psychology illusion. It's called the McGurk effect, and I'll let him just do it. He's saying ba.
Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba. Ba ba, ba ba, ba ba.
So that's weird, isn't it? It means that if you watch his lips, what you hear changes. So all of these three examples suggest in some way, based on our experience with language, our experience with faces, our experience with bicycles, we become hardwired. And this is a funny term. I'm sure you've all heard it. People my age are often criticized for being very hardwired. I want to give you a sense that this is more than just a metaphor. It actually is close to what the nervous system is made of. And if we go back to what Ellen said, it's related to the discoveries of Santiago Ramon y Cajal, who was a visual genius. He used a stain called the Golgi stain, which was a remarkably inefficient stain that labeled very small numbers of nerve cells, but he labeled them in their entirety. And this was due to an experiment that Camille Golgi, an Italian scientist, did in his kitchen with photographic emulsions and developed a stain that worked in the nervous system. And Cajal used this stain to great advantage to discover some fundamental principles I'm going to tell you about. It's ironic that although they shared the Nobel Prize, Golgi for the stain and Cajal for what he did with it, that at the Nobel Prize ceremony, Golgi spent his entire lecture explaining that Cajal was an idiot, that he had completely misunderstood what he saw. He was not seeing correctly. And it was a shame he did that because mostly what Cajal said was right. So let me explain what Cajal did. Cajal's big idea, which was based on the Golgi stain, is shown in this picture here. This is a drawing, one of thousands and thousands he made. As Ellen mentioned, there's an exhibit of his artwork, original paintings and drawings of the nervous system that's presently at MIT and will be there for a few more months. It's definitely worth seeing it if you want to see beautiful pictures. This is not one of the most beautiful pictures, but it makes the point that I want to make. It's a piece of the cerebral cortex of a cat, and these objects here are nerve cells that were stained with the Golgi stain, and then the paper he used has yellowed over time, but apparently he used a lot of whiteout to correct minuscule problems that no human being would ever have noticed were errors in the first place. It looks a little like snow on trees, but it isn't. That was not artistic. It was just the paper has not weathered as well as the whiteout. The key to this picture is not that there are nerve cells here, but these arrows. This was Cajal's great insight. He said, nerve cells have two kinds of processes coming out of them. They're very complicated cells. They have these little wires sticking out in all directions. One kind sends information to other cells. And here's one of those wires. It's called an axon. If it's leaving a cell, it's called an axon. As this arrow means information is coming from this axon. That cell body might be way over here somewhere. And that axon terminates in a bunch of branches that overlap the smaller processes of this nerve cell. These small processes are called dendrites, and they're like the antennas of a cell where it receives information. And Cajal inferred, just by looking at the Golgi stain, that this cell was imparting some information to the dendrites of this cell. The cell would take up that information and then send it out its own axon. And that axon would overlap with the apical dendrites of this particular cell. Apical because they're coming close to the surface of the brain, and that information then would flow into the cell body of that cell. These are where the nuclei are. And at the very same time, here's another axon from another cell that's contacting the basal dendrites of this cell, and together somehow these inputs are collaborating to generate a signal that then flows into the cell body and out the cell's axon. That axon touches the dendrite of this cell. That information then flows into that cell body and out, and that axon does the same here. And this is a remarkable insight because it didn't come from following anything over time, just by looking. He was a visual genius, and to a first approximation, this is the way we think about neural circuits today. Remarkable, and it had a big implication. This big idea had a big implication because you could explain behavior based on this kind of connectional map. And here's an example of Cajal's drawing of the way muscles might move if there is a stimulus to the skin that might be irritating, for example. So he said the skin might have processes coming into it from nerve cells. Information from the skin irritates that axon, which sends information in to the brain, and then that information flows out the axon to the terminal branches, where it is close to the dendrites of a neuron, and that information then goes from that cell to this neuron, perhaps in the thalamus. That neuron then sends information into the cerebral cortex and activates the dendrites of one of these pyramidal cells with apical and basal dendrites. That information then flows out that axon, goes all the way back down to the spinal cord, and activates the dendrites of what's called a motor neuron, a neuron in the spinal cord that sends its axons out to make neuromuscular junctions. And here you might have a loop of information going from sensory to motor through the nervous system. Now, this doesn't have to be the skin. If you drive a car and you're driving along and suddenly you see the light turn red, information gets sent to your right foot to take your foot off the gas and push the brake pedal. And if you're a little kid and you're in class and your teacher asks a question and you know the answer, let's say what 2 + 5 equals, and you're in first grade, information would be sent to your deltoideus muscle to pull your arm up and move your arm back and forth. So these kinds of wiring diagrams to do things like that could not possibly have come from genetics. Obviously, red lights are much more recent than our nervous systems, and yet somehow we have these pathways that are not cognitive. Once they're learned, they're sort of built in, hardwired into our nervous system. It would be nice to say that we've seen wiring diagrams like this, but we haven't. This is a dream. This was a dream from 1895. But to this day, we don't have wiring diagrams like this, and one of the challenges of laboratories like mine is to visualize these wiring diagrams to understand how they get instantiated, especially due to experience. So how might we see every single entity in a wiring diagram? The problem with the Golgi stain is it's very sparse, and very rarely would a whole network show up. So we thought one way to do this would be to have a technicolor Golgi. So instead of having a sparse stain that labels only a few cells dark brown, if every cell were labeled but each a different color, wow, then perhaps you could see the entire nervous system. So we made these transgenic animals. Josh Sains, I worked on this together, and generated these rainbow mice. Here's a picture of one of these rainbow mice. This is the dentate gyrus of the hippocampus. This is the rest of the hippocampus, and this is the cerebral cortex. And each of these cells is expressing a subset of fluorescent proteins. Fluorescent proteins have a lot to do with the Marine Biological Laboratory. Osamu Shimomura, who won the Nobel Prize for green fluorescent protein, did the seminal work that led to the discovery of this protein that you could genetically put into animals to make cells green. And then with the work of another Nobel laureate, Roger Chen, mutations of that fluorescent protein were made. So there was both a red, a green, and a blue fluorescent protein. And just mixing different combinations allowed you to have this extraordinary number of colors. So this looked really beautiful. And I'm going to show you one place where it really helped us. And that is the connections between nerve cells in the spinal cord and muscle fibers. The connections between muscle and nerve can be seen very nicely in rainbow animals where motor neurons, the neurons that activate muscle fibers, are labeled different colors. These neurons send their axons out into muscle, and if you look at the nerve coming out of these, it's quite beautiful. You can see the nerve comes out, and it's just lots of little wires. And to be honest, when I saw this, I was really excited because I realized we had developed a way to trace axons and wires over very long distances until a colleague of mine pointed out that computer scientists and people who build electronic devices have known this forever. Of course, color the wires, then you can follow them. So this is a form of interesting convergent evolution because you have both animals and machines made by humans that have the same fundamental property. Anyway, if you follow these nerves all the way to the periphery, you see the connections, the synapses these axons make on muscle fibers. And I'll just show you first a little region of this to give you a sense of the color depth. It's really beautiful. They're just gorgeous to look at. And if you trace these axons all the way to the periphery, you can look inside a muscle and see the connections these nerve cells make to muscle fibers. And these pretzel-shaped apparatus are neuromuscular junctions. They're places where axons are talking to muscle fibers. If you look carefully, you can gather that they look like they're clasping cylinders. And they are, because muscle fibers are cylindrical, and these are neuromuscular junctions that are placed around individual muscle fibers. And these colors tell us that this neuromuscular junction definitely did not come from the same axon as this neuromuscular junction, and this one came from a different one, and that one, and that one. You could map out, for example, in a muscle, every single neuromuscular junction. And I want to show you one such map that was done by a graduate student of mine, Julu, when he worked with me. Let's see here. So here is one of the axons to a muscle, and these little endings are those pretzel-like endings, but this is the entire muscle. This is one axon in the muscle, and here's a second axon in the same muscle, and here's a third. Now, I could show you them all at once, but I have a contract with Julu where he said it was so hard to do this experiment, you have to bring them in one at a time, otherwise it'll go by too quickly. So you're gonna have to see them one at a time. But don't worry, there are only 15 axons in this muscle, so it's not gonna take forever, but you have to see them one at a time. And it's interesting to see them one at a time because they each have their own kind of branching pattern, and it's kind of remarkable. Interesting, some of them have lots of connections, some of them have fewer. And I'm going from the axons that have the most synapses to the axons that have the least. We're almost done, don't worry. We have one more to go. So that is the entire wiring diagram, what you might call the connectome of a muscle called the interscutularis muscle in a mouse, which pulls the ears back. And so it's, yeah, but this was a lot of work. And as soon as he had a success, I said, great, now you have to do it on the other side of the same animal, and you've got to do a bunch of animals to see how stereotyped this wiring diagram is from one to another. Because you have the complete wiring diagram of this part of the nervous system. And so he did. And the result was, depending on your point of view, either very surprising or not very surprising, but it was that every single instantiation of this wiring diagram was different. The left was different from the right. The left of one animal was different from the left of another animal. There were no two that were the same. So that's interesting. Every neuromuscular junction is only contacted by one axon, so that was true in all cases. But the precise pattern here was different in every case. More disturbing, perhaps, is that there were lots of things in here that no engineer in their right mind would have done. There was a lot of suboptimality, useless loops, premature branching. Let me give you an example of this. This is one of Julu's reconstructions, and all the axons are labeled blue here so they don't get in our way, and one of them is labeled red. And I want to just focus on this region here. The axons are coming up from the central nervous system here. All of the axons go this way, except two. One of the two is an axon that bifurcates and goes both ways, but in this particular animal, this one axon, this is the only axon that only chose this path. And I am not a judgmental person. I don't know why it did that, but I accept it. But look what it does. It goes all the way up here, makes a hairpin turn, comes all the way back down here. If it goes any further, it'll leave the muscle, then it makes a hairpin turn again, and then it makes a branch out here. That branch innervates that neuromuscular junction. If it were me, I would have just made a little branch here and gone right there and saved a lot of this extra branching. But no, even worse, this neuromuscular junction is based on this bifurcation here, which seems reasonable. The axon bifurcates and sends a branch here, but both branches go up this way. Then they both come down here, and then one peels off, crosses over itself to go to that neuromuscular junction. Very, very disturbing. And things like this, you don't see this in invertebrates, highly evolved animals that have been around for a long time. But in mammals, we see evidence of this all the time. So where does this variability come from? Why are they all different from each other? And the answer is development. And this is surprising, but it is, I think, the key to, at least in my mind, to understand the purpose of wiring. If you look at an adult axon, and I've just shown you sliding in one after another examples like this. This is in a 2-week-old animal where each of these gray sites is a place where there is an axon making a synapse. This particular axon makes synapses on these particular muscle fibers. At 2 weeks of age, a human would be very, very young, but in a mouse, they're in their adolescence. They're teenagers already, you might say, at this point. They're going to be capable of having offspring 2 or 3 weeks after this. If we do the same study here of looking at one axon, not at 2 weeks of age, but at birth, you see in a mouse there is a radically different wiring diagram of each axon at birth. So here is an axon at birth, same muscle, and now rather than having, I don't know, 20 or so synapses, this axon has 10 times as many. There are some neuromuscular junction sites it doesn't contact, but the vast majority of the muscle is contacted by this one axon. And it's not just this axon. Every single axon at this age has this vastly more complicated wiring diagram at birth than it does a couple of weeks later. So we can measure, we can count the number of synapses they make at each of these ages, and when we did that, we discovered that the most complex wiring diagram was right at birth. And then over the first couple of postnatal weeks, especially the first postnatal week and a little bit in the second, there's a precipitous drop in the number of connections. So when it comes to the brain's wiring, at least in muscle, it's all downhill after birth. The brain gets simpler. Now, I think most of us would assume, well, we're adults, we have a very complicated brain, and babies, they don't know anything. They should have a very simple brain. It's just the opposite. Disturbing, perhaps. So what is going on? One thing to keep in mind here is not only is this axon branching to many more target cells, but if every axon's doing that, then this one neuromuscular junction site must be the home of many different axons, because the number of muscle fiber sites is not different between birth and here. So there must be a lot of fan-in addition to this fan-out. And to see that, we did a serial reconstruction of one neuromuscular junction. So this is a serial sections of muscle, shaved one section after another. We're just going through this block of images. This is the muscle fibers striations here. And then Juan Carlos Tapia labeled all the elements going to that neuromuscular junction. In the adult, I've already shown you there's only one axon going to a neuromuscular junction. At birth, it's a completely different story. There's a mass of axons. They're in a kind of happy state with each other. They're all touching, and they seem to be quite friendly. And there are, in fact, in this case, 10 or 11 different axons, all converging on the same muscle fiber. So a complicated wiring diagram at birth, many axons converge on one neuromuscular junction, and each axon sends branches to many neuromuscular junctions. It's as if every axon is trying out every potential target, and every potential target is evaluating every one of those inputs because they're all converging on the same site. You wait a week, most of the multiple connections are gone, but very often then you find muscle fibers where there are only 2 axons left. And that point, the two axons that are the survivors kind of square off. They each have, in some cases, half the neuromuscular junctions, and a competition ensues as to who's going to win and who's going to leave. And we could follow that over time with fluorescent proteins that are colored by imaging the receptors in the muscle in red and the two inputs at the same time. This is at postnatal day 11, and then just come back day after day and look at the same synapse. So that's postnatal day 11. Here's postnatal day 12. The axon that's yellow has gained a little territory. Postnatal day 13, axon yellow is getting more territory. Blue is on retreat. And by postnatal day 14, it's just axon yellow that has stayed, and it plumps up a little bit, but that's the end of it. So by following this process over time in a living animal, we witness the competition process by one axon invading the territory of the competing axon, which retreats. We call this synapse elimination because it finally is eliminated. Stability then reigns for the rest of the life of the synapse and probably the animal. These are stable until old age, with this monogamous coupling between a particular axon and a particular muscle fiber. I don't know which gender is which here, but there is some kind of relationship between the two of them, and that is it for this muscle. It's over, it's finished, and to summarize all this, you start off with a muscle with a very complicated all-to-all wiring diagram. The motor neurons each try to contact every muscle fiber, and every single muscle fiber is contacted by most of the axons. And then over a couple of weeks, you get to this hyper simple state where each muscle fiber belongs to only one axon and all the other branches have retracted. Every axon is a winner and every axon is a loser. But the final pattern is one where there's ownership or monogamous relationships, if you will, between particular neurons and particular muscle fibers. I'd like you to imagine this a metaphor for what youth is like and what an adult is like. Youth, you're exploring the world, trying out all sorts of things, and then a lot of those ideas turn out to be very terrible. Your parents told you they were bad ideas, you ignored them, but finally you become an adult, you settle down. So it raises the question, could it be that the same kind of pruning that's going on in the synaptic connections to muscle is also going on elsewhere, everywhere else in the developing brain? And let me put this in a slightly more scientific terminology. Could it be that we instantiate the memory traces as we learn about the world by sculpting our neural circuits via the process of synapse elimination in childhood? The thing I studied when I was a graduate student was this phenomenon in an autonomic ganglion, and I thought about it a lot, and as I thought about it more and more, it seemed to me the only explanation for what memory is like. This doesn't mean I was right, it just means the alternative ideas in my brain were pruned away, so that was the only idea I had left. If this is true, you might imagine that young children should spend a lot of time exploring the world because they have to build a wiring diagram based on exploration. And we don't think about babies as being exploratory particularly. I just want to show you a movie. If you have any doubts about the pathological level of curiosity young children have, look at this movie of a 9-month-old causing chaos in a dining room. 4 hours of play, but I'm going to show it to you over 2 minutes. And it's got music, so it's a remarkable, another piece of YouTube stuff.
Côte contre côte, nos corps vibrent partant. Et dans ces courses, du sang dérive, on tombe presque hors champ. Et ton terrible qui défonce plus que l'ego qui te prend. Tu nous réduis à l'impossible dans tes tourments. Et sans jet, tu te défonces, tu ne comprends pas. Car nous avions une belle histoire, tu le nieras pas. Par contre, tes cris défoncent les murs de ton appartement pour atteindre mon cœur qui meurt un peu plus à chaque instant. Car ensemble, rime avec tes hommes et l'homme que tu es n'est plus que discorde. Car ce que tu es rime avec tes grèves. Pour ma part, je n'ai que ce qui reste à jamais.
And it keeps looking at
Tête contre
very
tête, tu
interested
m'as
in
vécu
space,
juste à temps.
as you'll
Et
see.
quand tu es revenu, tu n'étais plus comme avant. Et tes mensonges, j'en ai que tu t'entêtes. Et sans gêne, tu te défonces. Tu ne comprends pas que tu détruis une belle histoire. Je le nierai pas. Par contre, nos cris détruisent les murs de ton appartement pour atteindre l'amour qui meurt un peu plus à chaque instant. Car ensemble, rime avec tes ordres. Et l'homme que tu es n'est plus que discorde. Car ce que tu es rime avec regret. Pour ma part, je n'ai que ce qui reste à jamais. Car ensemble, rime avec tes ordres. Et l'homme que tu es n'est plus que discorde. Car ce que tu es rime avec regret. Pour ma part, je n'ai que ce qui reste à jamais.
Gets trapped. So, the profound rewiring theory and its implications. Mature circuits of nerve cells, not just muscle, emerge after a period of pruning from the much more interconnected nervous system of juveniles. Pruning is mediated by neural activity, sensory experience, and motor practice that causes synapse elimination. We become what we become based on what remains after the pruning is over. We start out with the potential to be many things, and we end up, sadly, as a small subset of these. I'm particularly aware of this as a teacher, as a professor, because experience and even education in this sense is destructive. If this were true, and this is a theory, this is not a fact, in muscle it's a fact, but elsewhere I don't know, it would mean that babies are wired much more than adults, and there is, again, if you go to the most important data source of the world, which is YouTube, you find some anecdotal evidence that brains are overwired in young children, causing hypersensitivity to sensory stimuli. I'm going to give you just two examples here, and every one of these examples, there are thousands of examples of them. The first one I want to give you is that grass is very bad. I don't know why young children are stimulated in a negative way by grass, but they don't like it. And maybe it's because their sensory endings are connected to so much of their brain that the stimulus is just overwhelming. And so many children, if you try to lower them on the grass, they do this. This kid is going to try to touch the grass. Watch his face after he touches the grass. It's like he just bit into a lemon, but he's going to try again. Maybe. Yeah. And then look at his face again. Ugh. So grass is very bad for little kids, whereas meaningless sounds are very good. This should be. So we want to know what's going on in the brain. There's also one of ripping paper. Kids love that. Why? I don't know, but they don't like grass. So to try to see the brain, we wanted to use Brainbow for this, but the resolution you need to see brains, as you'll see in the coming up quite soon, you need more resolution than we can get out of Brainbow. So we approached this with electron microscopy that has much higher resolution, and we built a machine. I won't go through the details of it, but it's a machine that basically allows us to section the brain very thin and collect all the sections on a long piece of tape. And this is just a tape drive, putting tape up close to a block of brain tissue, and then we're cutting the brain tissue on a diamond knife. So here is the diamond knife edge, and the block is going up and down with a piece of brain in it, and it's sliced, and then the slices are going into a boat that's filled with water, and then one after another, here they are coming up, And we've dehumanized this part of electron microscopy. Dehumanized meaning getting the humans out of the pipeline. It goes a lot faster, it works all night, all day, and we can cut 10,000 sections that are each 30 nanometers thick, which is 1,000th as thick as a hair. And that kind of thinness gives us really wonderful resolution. Then we take this strip that has the brain that used to be a three-dimensional object, and now we've got it into very thin sections. We cut the strips up, we put them on silicon wafers, and then we put the silicon wafers in an electron microscope. Babby Kasturi, who helped develop this particular part of the technique, has very stable hands. So I asked him to hold his hand really still as we zoomed in on one of these sections of the brain. He is very brave. He's going to go into the electron microscope in a second, as you'll see. This is cerebral cortex, this is hippocampus. And these big white objects are blood vessels, and these smaller white objects are nerve cells. These big white stripes are the apical dendrites of nerve cells. These black outlined objects are myelinated axons. The disease, multiple sclerosis, is a problem for them. These dark things are mitochondria, energy organelles. And if we zoom up enough, finally, we see things that I recognize immediately, but I'm going to color them in to make it easier for you. We see dendrites, that's the receptive side of cells, and this dendrite has here a spine. They have lots of spines with a spine apparatus, and this is a cross-section of a thin section, of course, and the cross-section here has an axon on the other side, and the axon is filled with all these little circles. Those circles contain neurotransmitter, and this axon is going to release that neurotransmitter at this synaptic site. So good, that's the good news. We can see with this technique, using scanning electron microscopy, enough resolution to see synapses. The bad news is a problem of scale. That is that, and this is an image from work that Mike Greenberg and his lab and my lab have worked on. He's here this evening. The problem of scale is that to generate an image like this at that resolution requires to take image after image in the electron microscope, 30 images, altogether 5 hours to image one section, because you've got to take 30 tiles at very high resolution. And that's a problem, especially when you remember that at the end of all that, you have something this big, or actually smaller than that. It's about 1 millimeter on a side. And if you wanted to not just have 1 millimeter, but have 1 millimeter of depth, you would have to do that 5 hours 33,333 times to get 30 nanometer sections. I did the math for you, it's not pretty. At that imaging rate, a minuscule box of 1 by 1 by 1 millimeter would take 17 years. I've had a hard time attracting graduate students to my lab. Postdocs, not as much of a problem. They kind of like this idea that it's going to take forever. But graduate students want their mentor to be alive when they're graduating. So we have to be able to image the brain a lot faster. And I wouldn't be asking if we can do that if we couldn't. Thanks to this company, Zeiss, we have a device that they built in collaboration with us to make this possible. So instead of taking 17 years, we can now do it in 6 months. This makes me semi-happy for several reasons. One is I'm standing on a chair with wheels. This is gigantic. It's like a refrigerator for giants. It's just huge. I'm not the tallest person in the world, but my hand is over my head, and I'm holding on for dear life. That's one reason. The other thing is, this is called the Multisem 505, which gives you the impression there was a 504 or a 503. This is the first one, and this was there. I think that 505, I don't really know why they call it 505, but I think it was for me to make me feel more reassured that they knew what they were doing. And it took some time to get it up and running, but it is now an automated image acquisition device for getting wiring diagrams, connectomics. We can generate 11 terabytes a day using probably the fastest electron microscope ever built. Speed is the issue. I won't go into much technical details about it. It's a remarkable piece of engineering, and the magic to it is instead of scanning one section piece at a time, it has 61 beams, all scanning simultaneously. So what took a long time before can now be, like an hour, now can be done in a 60th of an hour, like a minute. I'm a biologist. I don't know math. I had to think that through. Okay, so I'm not going to go through the details of this, but let me just show you what images look like. When you take them on this, it's really a beautiful machine. The resolution is great. And I'm just going to show you a stack of images where we're going to look at the tape, one section after another, that's been aligned. And this is what brain looks like, this piece of cerebral cortex, just going through a stack. This is a stack that has several thousand sections in it. This is the nucleus of a neuron. That's the nucleus of another neuron. And it looks like everything's moving. That's the Golgi apparatus, mitochondria. Things are moving in space. This is like a space lapse, not a time lapse image. We're just cutting through basically a volume of brain. And so these objects that we're seeing are just moving around because the objects themselves are running at some arbitrary angle relative to the volume. You don't fully appreciate the complexity of the brain. You do see there's a lot of stuff between these nerve cells, but you don't appreciate it until we zoom up to the native resolution. So when you look at native resolution, you can see that there's just a gazillion wires running around. Every once in a while, the wires will have dots in them. Those are the synapses. And anyone can basically follow an object from one section to the next because they're contiguous. These are wires that are running through our volume. I'm just arbitrarily picking one out, and there's no problem following that. Any person could follow it. And in fact, you end up with a digital tissue, a box of brain, if you will, where now every bit of the information at the not the molecular level, but at the structural level is available to you. And then you can figure out what the wiring diagram is by just using a coloring book strategy. So Daniel Berger built a little program that's called VAST, that's used by a lot of labs now. That's basically a digital coloring book where you find an object like, say, a dendrite, you color it in again on your computer screen. You don't need crayons for this. Even undergraduates can do this. Second graders can do this. Anybody can do this. And anyone who wants to, we've got plenty to do. So here's a dendrite with dendritic spines, and here's an axon that makes synapses on 2 of those spines. That comes right out of that. So the data is good enough to reconstruct everything, and you'll see here there should be vesicles in this cross-section, of an axon making a synapse on a spine. And then you don't just want to do that, you want to do everything. And so here is a manual segmentation of a whole region where every single cellular element in this piece of brain was reconstructed. And we did this because we wanted to see within a dendritic spine distance of this gigantic red dendrite running through, what else is there? We wanted to itemize every single item. And we did that for this red dendrite and this green dendrite right next to it. I want to just show you those results. So here are those dendrites. We did twice the length of the red as the green. They have lots of spines. But in this area, there are other dendrites that are pushing into the same area, and lots of dendritic spines of other cells. Here's all the dendrites in this volume. And that, by the way, is the spine that sticks out the furthest. You'll see why I mentioned that in a second. There it is. So this is the red dendrite and the green dendrite. Here's all the other pieces of dendrites that are within a cylindrical distance of the longest spines from these 2 cells. So lots of dendrites squeezed in there. Of course, these dendrites are being contacted by axons, so there also has to be room for axons here. Here are all the axons. It almost seems impossible that they're both in the same place, but wherever there's a dendrite, there's a space, there's no axon. And where there's axons, there's no dendrites. And there's also supporting cells called glia. Mostly there are astrocytes in there as well. Here's everything, and they're sticking out that little dendritic spine. And then what we did is we went ahead and analyzed each. PowerPoint has stopped working. PowerPoint is restarting. Hey, sorry. This will just take 1 second, I assume. We don't have to go through the bicycle again. 1 second here. Here. All right. So here is all the things in this little area. And by virtue of imaging, we could itemize every single one of these objects. And this is either depressing or very depressing. I don't know. There's just way too much stuff here. This is ridiculous. We're just looking around the dendritic spines of one cell and a neighboring cell. And you could classify it, and this will just separate them into the main categories. There's axons, glia, and dendrites. And the axons can be separated into myelinated ones, inhibitory ones, excitatory ones, oligodendrocytes, astrocytes. Embarrassingly, a bunch of things we still don't know what they are. Spiny dendrites and smooth dendrites. That all seems good until you realize that's what we did. These are two neurons. That's the area we reconstructed. So I never usually show it that way. I just show it this way. It looks big, but it really is a lot of work for the complete rendering of almost nothing. And in there are 1,400 different axons, 193 dendrites, and 1,700 synapses. But you can see every synaptic vesicle. You can see everything. So here is for the red dendrite, all the axons that are making synapses on it. And there's a lot of biology that I could talk about here that I'm going to not bore you with as we move on to what we take this to mean is a proof of principle that we now could do this on a large scale, especially if we could automate the remaining tasks, like the coloring in. And we have begun doing that to great success, thanks largely not to us, but to this company, Google. So here is what we're doing now. It's a volume that's 10 million times larger than the last volume, which took 5 years. So this should, if we did it by the way we were doing it before, should take 50 million years to finish. But they're already doing this so fast that it's taking my breath away how they're doing it. So this is a piece of human cortex from a living human being who was having neurosurgery for epilepsy, and a piece of the medial lobe of the temporal gyrus was removed so this neurosurgeons could get the tools in to remove the lesion site of the epileptic focus in the hippocampus. And they said it's a very small piece of tissue they're giving us, but it was actually huge, and we had to cut it down to make it possible to image. It's 2 millimeters in this direction and 3 millimeters in that direction, and we image this with the multi-beam. So this is 500,000 pixels this way and 751,000 pixels that way. To give you and we have 5,000 sections. So this image itself is 375 gigabytes. And if you multiply that by 5,000, you have 1,850 terabytes, or 1.85 petabytes, which is such a big number, nobody knows what that means. So here is a number, everybody knows what a megabyte is. This data set is 1.85 billion megabytes. So this is a big project, and Google likes projects that are hard, so they wanted to help us with it. And what they have taken advantage of is artificial intelligence to train their classifiers to do all the wiring automatically. And so far, they've only done this region, but it's part of the training process. And this is the region they've done using artificial intelligence and the massive computer infrastructure that Viren Jain and his group has access to. I mean, millions of CPUs and thousands of GPUs, and a very impressive segmentation algorithm that's going to come out in Nature Methods. So this is a data set where they have reconstructed everything for us, where I got this back yesterday, actually. This is the most recent one back. And I don't know what this looks like. It doesn't look like, it looks like rainbow as I would have imagined if everything could be a different color. But this is done with the electron microscope. And it's remarkable because you just go in and click on any particular cell, it lights up, and then you just double click on it and it will render the three-dimensional version of each of those things, including all the dendritic spines. So it's just remarkable. So it's the rainbow equivalent of electron microscope, but now with much higher resolution. We'll soon have, I believe, the complete neural circuit of a slab of human cerebral cortex with more than 100,000 neuron cell bodies in it and billions of synapses. This, of course, is a minuscule part of what's even in this little box I just showed you, which is itself a minuscule part. It's only 5 terabytes of something that's going to be 1,800 terabytes. And this is so complicated, it brings to mind my last slide, which is There's a crisis coming in every field, but especially, I think, in biology, and it's related to the relation between information and understanding, the great complexity problem coming at us. In most laboratories, when this lab head begins to talk, they say, I'm seeking to understand something, and we take it for granted that we want to understand things. In my lab, we've banned the word understanding, because when I was a student, understanding was very straightforward. We had a lot of pseudo-understanding. We had ideas that had great power, and the one thing that never stood in the way was actual data. And you can see what's happening now. We now have a glut of information, and I believe the biggest casualty of this big data may be the big ideas that got us where we are. I want to end with that problematic note and remind you that I did almost nothing. This was done largely by my lab colleagues and collaborators and support. Thank you very much. Happy to answer some questions.
Okay, I'll just say now, I'm sure there are about a gazillion questions, but we'll just have time for just a few before we go over to the MIGS room.
Jeff, that was magnificent, but my question is, what is the answer to the question in your title? Can the brain structure reveal its function?
Yeah. Good question. So my view is that if it turns out that the way humans instantiate information is by pruning circuits, there must be some rules to the way those pruning events occur. And that even though in a little piece of brain I couldn't tell you possibly what's being learned, I think in a cortex of a human at least, a lot of that stuff is to store information. And if there are any irregularities to the way information is stored, given the fact that we're going to know everything about the wiring diagram, down to every last synapse, every last synaptic vesicle, I think we will see some structural motifs that must reflect in some way the meaning. It very much the way I think of genomics. Let's say you didn't know about the triplet code, or you didn't know about anything other than the fact that DNA had heredity in it. If you had a tool to just reconstruct every single nucleotide, and that was the first thing you had, I don't know, but I suspect eventually a lot of regularities would come out of that that would lead you in the direction you'd like to go. And I think connectomics is in a similar boat, basically.
I want to ask about something at the beginning of your talk. If you cut a peripheral nerve, the axons will regenerate back to where they were originally. I don't know if you've had a chance to look at that situation, but I'm wondering whether the regenerated nerve recapitulates the complexity and then the simplification that occurs in normal development.
Yes, it does. You go through a period, again, of extra elaboration of branches and synapse elimination. Interestingly, if you crush a nerve, not cut it, but crush it, the axons will cross the crush site and reoccupy the former Schwann cell tubes, and you'll even reconstruct the wiring diagram. If you cut the nerve, or you double crush once and then crush the second time, the wiring diagram the second time around is distorted relative to the first. But in all cases, when axons grow back, they branch and make extra branches, usually at the nodes of Ranvier, and make extra branches to innervate muscle fibers. Then you have these competitions again, and you're down to one, in most cases.
Hi, that was a fascinating talk. I have so many questions, but I'm going to ask one. So there's a psychologist named Jonathan Haidt, and one of the theories regarding what you showed us is that the rise of anxiety disorders in teenagers is a result of parents not allowing children to play when they're young. So to what degree is that true? And has there been any study on population which, like, seeing the brains of teenagers who have not, got that chance to play it when they're young?
Yeah, obviously getting human brain tissue is a new thing and problematic, especially from living people. If you can get postmortem tissue, but it's not fixed very well. In terms of anxiety disorders, it wouldn't shock me if there is some kind of wiring problem, but whether childhood play has anything to do with this is so far out of my own knowledge, I wouldn't even dare guess. Play on the graph. I mean, this thing, my daughter gave me this YouTube on the grass playing, and I remember reading many years ago that children who have autism or the autism spectrum disorders have a prolonged period of inability to walk barefoot on grass, which is interesting because some people think that is a disease, that is a pruning abnormality.
The picture of the 9-month-old baby playing for 4 hours that was sped up. How could this baby go without anything to eat or drink or take a rest or anything?
First of all, I had nothing to do with that. I just found it on YouTube. I think there is a moment in there where it stops. I think the kid either gets a diaper change or something. I mean, 4 hours. Yeah, good question. That kid's going to have anxiety later on.
Jeff, up here. Hi, Jeff. This is Bill Chin. How are you doing?
Good. Hi, how are you?
Great talk. So in terms of pruning, you talked about maybe 2 time points. So question is, any chance to look at an aged animal? And really more to the point, are there any diseases that you think are known, either genetic in animals or in humans, that have either under pruning or over pruning?
I mean, there are animal models where synapse loss does not take place in muscle. They're not that usual, and in most cases, they're diseases where the muscle fibers themselves are not firing action potentials very well. So these animals are very weak, but the activity-dependent plasticities, whatever they are probably not working normally. In humans, there's lots of discussion whether synesthesia is an example of multimodal connections that should have been pruned that stay. Some people think schizophrenia could be a disease where there are connections that shouldn't belong that make you think when you hear your internal voice that it's an outside voice. I have no idea about these. I don't know what kind of abnormalities one would expect. I think there are a lot of children who fail to thrive, who stay in an early developmental state, never really learn to walk. It would be interesting, I guess, to assay whether these kind of processes have not been going on. But honestly, we're just at the very infancy of this kind of work.
Okay, I'll ask it.
Where?
Over here.
Over where? Over there.
Back in. Thinking about your mouse models and your now ability to kind of map at early stages and then after pruning, has it given you any insights to the old debate of sort of nature versus nurture? And to what extent is genetics mapping out the neurons ahead of time and then the pruning from the environment? Have you done those studies or looked at maybe clones and that kind of thing?
No, I mean, we've certainly thought a lot about this. There's a tension, I think, between the triumphs of molecular biology and the power of molecules to generate neuronal phenotypes and synaptic plasticity phenotypes, which could give the impression that the whole thing is sort of preordained based on genes, but no one would say that a human stepping on the brake pedal when they see a red light is because you have a gene for the red light. There's no red light in your brain. So there has to be both nature and nurture here. I guess the question is, with something like the cerebral cortex, which is rather uniform, not very uniform, but rather uniform as a tissue, whether it really matters exactly how the cells are arranged. A lot of people say, absolutely, we have to get the cortical circuit because that will teach us how it works. It is possible that once you have a learning rule, it wouldn't even matter what you have as the substrate. You may know that children who have tumors or other problems in the cerebellum and get cerebellar removal at a young age go on to develop good balance based on cortical mechanisms. As if the cortex could do that, and also people who don't use their visual cortex, the auditory cortex apparently takes over some of the visual cortical space, suggesting again that although there is a defined area for audition and a defined area for vision, it's more of a multi-purpose machine, a learning machine, if you will, that is ordinarily divided roughly the same from person to person, but that may not be the essence of what's going on. In muscle, certainly it doesn't matter which particular axon touches which particular muscle fiber. What does matter is that every muscle fiber end up with one input to give you a graded kind of contraction. So it may be something, you know, the rules may work in a way that the nature part is at the moment so predominant, it's very hard to get a word in edgewise for nurture, that maybe the pendulum will swing a little the other way, but I don't know.
So this is a, oh, sorry, I'm right here.
Okay.
This is a pondering sort of when I very much enjoyed everything you had to say and how you shared it with us, so thank you. This makes me think of Timothy Leary, it makes me think of peyote and ayahuasca, and I just wonder, are there this thing of...
Timothy Leary, I know. Who are those other people?
Well, those are drugs, yeah. You probably shouldn't act like you know what I'm talking about there. But just this desire and somewhere inherent for some people, and knowing that they want to go beyond this pruning, they refuse to have those pieces minimized. And so it just made me think of that. I wanted to bring that up.
I believe I'm a very plastic, open-minded adult. My daughter is sitting here. She would probably tell you otherwise. And I remember feeling that my parents had become more predictable in their behavior. I'm not sure Timothy Leary could be otherwise. I think because of his mind-altering experience as a young person, his brain was wired differently, so he was a weird duck the rest of his life. But I'm not sure that that weirdness was constantly changing. I think he was a particular weird duck till the day he died.
All right. Thank you, everyone, and please join us in the MIGS room.